Hi everyone, today we'll be talking about the cell cycle and mitosis. Do you ever wonder how a small puppy can turn into a huge dog? Well, the answer to that question is cell division. How does a sperm and an egg turn into a baby? The answer to that question is also cell division. So the cell cycle is the life cycle of a cell from its formation from a parent cell until its own division into two daughter cells. This is what the cell cycle looks like. The four phases are the G1 phase, the S phase, the G2 phase, and the M phase. So G1 phase is where the cell grows and performs its normal functions and also makes the enzymes and nutrients it needs for the next step, which is DNA replication. During S phase, the cell replicates its DNA. During the G2 phase, the cell continues to grow and starts preparing for the next phase, which is mitosis. The M phase is when mitosis occurs, and that's when a parent cell divides into two identical daughter cells. There's also the G0 phase, or the resting phase, and cells that enter this phase do not undergo cell division. Some examples would be neurons and heart muscle cells. So the G1, S, and G2 phases make up what is called interphase, and cells spend most of their time, around 90%, in interphase and only 10% in the M phase. Let's move on to cell cycle checkpoints. There are three checkpoints that you'll need to know. There's the G1 checkpoint, the G2 checkpoint, and the spindle checkpoint. At the G1 checkpoint, the cell checks for cell size, the nutrients that are needed for DNA replication, growth factors, as well as DNA damage, since the cell is going to replicate its DNA in the next phase. It's also important to note that the G1 checkpoint is where the cell decides whether or not to proceed with division. The next checkpoint is the G2 checkpoint, and this is where the cell checks for DNA damage as well as DNA replication. And this checkpoint is really important because if the cell detects any DNA damage, it will pause and fix those errors before actually entering mitosis. The last checkpoint is the spindle checkpoint, where the cell checks for spindle microtubule attachment to the chromatids, and this checkpoint is also very important because if the spindle microtubules aren't attached correctly, then it could lead to non-disjunction and other serious problems. Now let's talk about cell cycle regulators. Cyclins are a family of proteins that controls the progression of a cell through the cell cycle via the activation of CDKs. It's important to know that different types of cyclins are only present at certain phases. So for example, the concentration of the S cyclin begins to increase during the S phase, reaches a peak at the G2 phase, and then decreases during the M phase. Next is cyclin-dependent kinases, or simply CDKs. Kinases are enzymes that phosphorylate other proteins, and CDKs are a family of protein kinases that works with cyclins to regulate the cell cycle. So when a cyclin isn't bound to a CDK, it remains inactive, but when a cyclin does bind to a CDK, it becomes active. Once activated, the CDK can phosphorylate or add phosphate groups to target molecules. The maturation promoting factor, or the MPF, is the most common example of how cyclins and CDKs work together. Once the CDK is activated by an M cyclin, it can phosphorylate other molecules and stimulate the cell to enter mitosis. Mitosis is a type of cell division that results in two identical diploid daughter cells. So before I move on, I wanted to talk about haploid cells and diploid cells. So diploid cells contain two complete sets of chromosomes, while haploid cells only have one set of chromosomes, or half the number of chromosomes as a diploid cell. It's also important to know that mitosis only occurs in somatic cells, and a somatic cell is any cell in an organism other than the reproductive cells. A helpful acronym to help you remember the steps of mitosis is PMAT. The P stands for prophase, the M stands for metaphase, the A stands for anaphase, and the T stands for telophase. During prophase, the nucleolus disappears, chromatin condenses into visible chromosomes, the centrosomes separate, and the mitotic spindle forms. During metaphase, the chromosomes line up in the metaphase plate. And then during anaphase, the microtubules that aren't attached to the chromosomes help elongate the cell, while the microtubules that are attached to the chromosomes shorten so that the chromatids can move to opposite poles. During telophase, the new nuclear envelope forms, the chromosomes go back into chromatin form, the nucleoli reappear, and the cell continues to elongate. And then lastly, cytokinesis is when the cytoplasm divides to form two daughter cells. We are talking about a cycle here, so once the cell is done with M phase, it re-enters interphase. So what's the point of mitosis? Well, the first purpose of mitosis is to produce daughter cells that are identical to the parent cell. And the second purpose is to maintain the proper number of chromosomes from one generation to the next. I wanted to end this video by talking about cancer. Cancer is a disease caused by the uncontrolled division of abnormal cells in the body. It can also be described as mitosis out of control. In order for a cell to become cancerous, it has to override cell cycle checkpoints as well as grow in an unregulated manner. And this is often how tumors form. In order for a cell to become cancerous, it also has to avoid apoptosis. A simple definition of apoptosis would be programmed cell death. You might be wondering, how does a cell even get to that point? It's not likely, but it is possible that you might get a question related to this on an FRQ. A quick answer to that question would be that a mutation occurred in a protein that normally controls a cell's progression through the cell cycle. Without regulation of the cell cycle, the cancerous cell will keep dividing and not undergo apoptosis. In this picture, when a normal cell detects cell damage, the damaged cell will undergo apoptosis. However, in a cancer cell, even when there is cell damage, the cell will keep dividing because its cell cycle isn't regulated, and this can lead to tumors. Here's a quick summary before we end. So in this video, we've talked about the phases of the cell cycle, which include the G1 phase, the S phase, the G2 phase, and the M phase, and how a cell spends most of its time in interphase. Next, we talked about cell cycle checkpoints, which include the G1 checkpoint, the G2 checkpoint, as well as the spindle checkpoint. Then we talked about cell cycle regulators and how cyclins and cyclin-dependent kinases work together to control the cell cycle. Next, we talked about mitosis, which is the splitting of a parent cell into two identical daughter cells, and its different phases, such as prophase, metaphase, anaphase, telophase, and cytokinesis. Lastly, we talked about cancer and how cancerous cells divide rapidly due to the fact that their cell cycles are unregulated. Thank you so much for watching this video, and I hope it helped you understand the cell cycle and mitosis a little better.